Então, nós vamos dar início à próxima palestra. O professor Tarik Meziane, do Museu de História Natural de Paris, com o título Sources and Fates of Organic Matter in Amazonian Waters and Elsewhere. Tarik, please.
Well, thank you. I'd like to start by thanking Fabio for inviting me to this conference, which is very interesting the way how it is done. A quick look for my institution, which is the National Museum of Natural History in Paris, which deals mainly with past, present, and future bio and geodiversity. But dealing with biodiversity, this institution is also doing active science in many fields, including aquatic science. And we have a lab there called Aqua, which deals with this subject. And we had the opportunity with Gwenaël Abril, who talked to you earlier, to work on the Carbama project, in which we were very much involved. And what I'm going to present to you today is mainly works that has been done during this Carbama work. And I put an elsewhere. It's just a few examples that I found interesting to add to this general talk. One which deals with the fishes in the Amazonian. It has been done by my colleague Marc Pouilly. It's a work within the main team, our team. But I hope you found it interesting. And other small piece of work to show you how studying organic matter through tracers can be rewarding and interesting in terms of ecological significance. So let's talk about the source and fate of particulate organic matter in the Amazonian waters. Which was interesting is that Gwenaël, who introduced to you the whole objectives, also the field work and the field study. Briefly, we were not involved in every single 8 of his cruises, but we did go in 4 of the cruises. And in these cruises, basically, we covered the high water, the low water, at almost low water, the falling water, and the receding water within 2 years period. And we samples along the Amazon River and the narrow rivers and Solimões, but also in Varzeas, mainly in 5 of the Varzeas. So we did sample in each of these territories. We did sample many, what we define as sources. And these sources include, of course, the suspended particulate organic matter, many aquatic plants, as much as we could have, and we also looked at the phytoplankton, sediments under the water, and also soils on the margin of the rivers and the Varzeas. So we pick up all these samples and we did many analysis. And what I'm going to show you today is mainly the analysis through fatty acid markers, a method that we used and developed in my laboratory, and also stable isotopes. And you will see later on in the degradation experiments that accompanied all this field work, how we can use other type of markers. And starting from here, maybe a quick glimpse about fatty acid analysis as a tracer of the organic matter. The experimental setting is you extract total lipids, you make saponification by taking away all the fatty acids that are in the membranes, that you collect them all, and then you methylate this fatty acid, and it's under this methylation aspect that it can be identified using GC and of GC-FID, and of course GCMS for name identification. So fatty acids are written in this form X, Y, W, Z, where X is the number of a carbon, Y is the number of double bonds, and Z is the position of the ultimate double bond. And you get this kind of graphic here, and each peak represents 1 fatty acid, say this one, and the surface of the peak represent the contribution of the fatty acid, the relative contribution of the fatty acid to the total fatty acids. So, for example, some fatty acids like these branched fatty acids here, iso and anti-iso 15, but also other branched fatty acids can be used as, and I explain you how, tracer of some sources here in this case bacteria. Others polyunsaturated are tracers of microalgae, others are tracers of phytoplankton, and so on. Long chain fatty acids can be tracing the of course vascular plants, mainly terrestrial one. But in our field we have to concert about that, so that means we need to identify our own sources of fatty acids in the environment we study. Of course, we also we did unstable isotope, but I think I will skip this part. I assume that everyone knows about the biplot of azote and carbon. C3 and C4 leaves, roots have different signatures and within the same plant. In this case, you see that the biplot signatures of the roots are different than one from the leaves. This is a C4 plant, for instance. This fractionation, the sources are used by consumers, but we assume usually that the fractionation is about 1 per mile for the carbon, 2.5 per mile for the nitrogen. However, recent works and there is more evidence to say that this fractionation has to be carefully used and needs to be adapted to the environment. For instance, in some mangrove ecosystems, the fractionation for first consumer will be not 1 per mile for the carbon, but exactly 5.45. Just to, this is what usually people use, but not necessarily the best way to use the fractionation. Above these technical issues, usually go back to the fatty acids. What you get is these huge tables with all the sources, with all sampling in one table. You make a Bryer-Curtis similarity matrices or any one that uses you, but we're using percentages here. So Bryer-Curtis is the adapted method, and we get this kind of results. This non-metric multidimensional scaling using the primer software, and what you have here is a plots of where you can see the contribution of the one particular fatty acids, because in each points you have the whole fatty acids, sometimes more than 60, but you can focus in one of them in particular. And the similarity distance between sample, closer the points, more similar are their fatty acid profiles. So that's about the distance, and then you get the circles that shows you the contribution of each fatty acid within the sample. So here we had about I think 200 samples, all made, and you will see from where they come. So here the results are about the spatial and seasonal variability of organic matter quality in the few Amazonian water we have looked at. As I said, we have to identify clearly the sources of organic matter through the fatty acids. And for example, for the cyanobacteria, the soup of cyanobacteria that we get at low waters, you see that this 16,1 omega-7, this monounsaturated, is very high compared to any other fatty acids. That macrophytes shows a very huge contribution of these two, 18,2 omega-6, 18,3 omega-3, which are very important fatty acids because they can be precursors for many essential fatty acids on the food web, for instance, and they are very important on leaves, also a little bit on the roots, but still they are completely dominant in the leaves of these macrophytes, especially in C4 macrophytes. Of course, we also looked at the soils, and you can see that the total of the saturated fatty acids here is more than 10%, which is, when you do ecological studies on fatty acids as tracers, it's huge, it's big. I mean, 10% of long chain fatty acids is not unimportant, it's very significant. That's what we find, for example, in some soils and the margin around the lakes. Quick glimpse, and it was interesting to hear the talk of Gwen when he told you that during the falling water, that's the time where you got many fruits and leaves coming from the terrestrial zone or the flooded forest coming to the water. Well, more or less that's what we can see here, and in this plot what we have is the five lakes that we have, Varzeas, that we have analyzed from Kabalyana, Janawaka, Miratuba, Kanachari, and Kurwai. And here we have the total fatty acids, which is a kind of proxy of the biomasses that are present in the suspended particulate organic matter. We are doing this in water. And then we looked on particular fatty acids, the one they mark cyanobacteria, macrophyte, and also the terrestrial one that I was showing you. And you can see clearly that the falling water, first of all, compared to high water, that the total fatty acids are increasing. Still the case for low water, and then go back to, I would say, baseline during the receding water. And what you can see also, if you look well, but we have here kind of a gradient from Kabalyana to Kurwai, but it's mainly because Kurwai is a very special lake, and it has, it seems, fatty acids contribution that is quite different from the other lakes. Also from the microphyte point of view, cyanobacteria, and even for terrestrial, it's terrestrial markers. So you looked on the sources, you look on the original of the markers, you look on the SPOM, and that's kind of details you can have, and that may help you to explain sometimes the dynamic of the organic matter and how it can impact, for example, carbon fluxes. If you look in Varzeas this time, just on the Varzeas, go back to the graphics you see here. This is the non-multidimensional scaling. Just realize that the white circles are the white rivers, the gray are the clear black rivers, and in black dot these are the waters that were analyzed from Varzeas. It's all pooled, and if you look at the difference just between high water and low water, you see that, for example, for the marker of cyanobacteria, if you look on the Varzeas, from high water to low water, as I explained you, the contribution of the cyanobacteria is hugely increasing compared to high water. Here, this all black points. And the other example will be the terrestrial plants, where you have a high concentration, for example, not high, but equivalent concentration from high to low in all water together. Aquatic plants, you have a better also concentration of their markers in low water than in high water. So roughly we can say that the composition of Varzea suggests the contribution of autochthonous organic matter derived from cyanobacteria and aquatic plant, because it increases, but still terrestrial markers are present and may indicate that contribution, even if it's probably lower than the others. Of course, this is not a this is a general view. Here again, it's not lake by lake. And if you look at the stable isotopes this time, so we are looking at the same samples. It has been varied analysis. So for each sample has been separated and analyzed for fatty acids and for stable isotope. And if you look at that, and if you do the biplot of carbon and nitrogen, and you can clearly see that the SPOM, the soil, you see that the main contributor to the carbon would be C3 plants here. Okay, and the C4 plants is quite derived to the right there here, and the C4 plants seems to not at all contribute in terms of a stable isotope to the whole package of samples that we have analyzed. So we have a significant depletion of nitrogen from high water to low water, because this is the difference: high water, low water. And this also may indicate that the atmospheric nitrogen is of course fixed by cyanobacteria. The question mark doesn't have to be here, in fact, but it's for sure. That's the two results we can have them from the stable isotope, and that can add to what we have seen in fatty acids in terms of contribution of markers. So, for example, the fatty acids of microphyte would be coming mainly from C3 plants than C4 plants. They share this kind of markers. So the stable isotope gives us better definition in that particular case, segregating between C3 and C4 in terms of contribution to the organic matter in the SPOM. That's in the Varzea. So what's happening here is that in high water there are large areas. It's very shame. I mean, I'm always respectfully saying this to many of you here in this room. I know they work in Amazonia. They know all the story. But large areas of Varzea lakes covered by aquatic microphyte. There's a low plankton biomass in the low water. There's less, of course, water, and therefore the plankton maximum standing stock. We have it. We can see it with all the tracers, and there's also resuspension. And we got intensive degradation processes on microphyte. This one also we can see it with the active bacterial markers present there. So we have accumulation of plant detritus also in the ecosystem at low water, and we also can have we can see that this phytoplankton is dominant at low water by the fatty acids and by the depletion also of nitrogen. In the river this time, from high water to low water, we also see increasing concentration of phytoplankton. So the rivers, white rivers and clear and black rivers in gray, and we can see that there is also a high increasing concentration of phytoplankton and aquatic plant markers, which also may suggest a transfer. I insist that suggests a transfer of organic matter from the Varzeas, and that can be the case during in particular time of the year. So now that we saw that we can mark the organic matter and the sources in this watering by some tracers, we were interested, and even if eventually it wasn't the case at the beginning of the project, but we were thinking, as we are aquatic ecologists, to see how this organic matter is conserved and transferred to upper on the food web. So the fish in Amazonia have been told they're very good to eat, as suggested by this picture. So let's see what this fish eats, in fact. And we did a work it's here, sorry. We did it in some mini lakes. We mainly actually 2-3 lakes, and we sampled fished some piscivorous, so carnivorous fishes, non-piscivorous, non-insectivorous, and other herbivorous, strict that eat only plants. That's what we know about them, and other that are more like omnivorous and even detritivorous. We looked on the detrivorous because we had this assumption that root system is so strong over there that we may need to look how these roots could be used. So looking at these fishes, we in fact extract fatty acids and looked for stable isotopes in each of the species we have studied here. And here we get is the fatty acids plotting of non-multidimensional scaling. And more or less we were able to group for at least 4 main groups here, which are the piscivorous, the detritivorous, the rutivorous. Doesn't exist the word, but let's call it like this. Fishes that are supposed to eat only roots, here in blue, and also the frugivorous. These 4 groups were statistically quite significantly different to each other compared to other little groups like omnivorous, fishes that eat algae, and so on. So we were able to group this species. Remember, each points represent the whole fatty acid composition of the species of the tissue. So each point is representing the averaging more than 30 to 40 fatty acids. That's in general. If you look on the composition, how this is, we get it. Now if we look in a little bit more detail of particular fatty acids that are known to be tracer, and for instance, if you remember this 18,3 omega-3, 18,2 omega-6, the one we say that are tracer of aquatic plants, you can see, for instance, that the tendency here, and of course they are present everywhere, for the simple reason that these fatty acids are essential for the living of the animals. So if they get them not directly from a source, they will eat them by, if they are piscivorous, by eating other fishes that accumulate these fatty acids. still they need these fatty acids to deal with the needs and the basic needs of physiological needs. So you see, for example, for the frugivores, here for this at one of these two fatty acids, the big concentration, which of course can be surprising, but yes, that's what it says. That says that this 18 to omega 6 is the fatty acid that more or less is dominant and representing the sourcing of organic matter in this group of consumers. We also have phytoplankton fatty acids, the one we found very important in fatty acids in phytoplankton, in the soup of the phytoplankton also, so the 22,6 omega 3. There is no clear tendency between all the groups, but more or less you see that the piscivorous has this big amount of these two fatty acids. So there is a mixed source for each of more or less each of the group, with tendencies that let each group a little bit specialized. But what we have here, which is less we are more or less confident than in other fields, like in for example in estuaries or open water, is that there is no significant tendency for one particular marker to say this animal eat only this kind of source. They are quite plastic. There is quite plasticity in feeding in getting their organic consuming organic source. What we need to know is that, for example, for this group, these detritivores animals, here they have quite important 24 omega 6, which we know is present very much in the plankton. And this animal, because we know it's the dent that he has, his behavior, this species shouldn't be able to eat directly phytoplankton, of course. So we assume, looking at the fatty acids, that there is a trophic link, that they had to eat something. These detritivores, so they had to eat an intermediate, for example, zooplankton or any kind of periphyton microalgae that lives on the detritus, and so on. For the piscivores here, with this 26 omega 3, which is very much present in this group of animals, this 22,6 omega 3 cannot be, of course, they're carnivores. So to get them, they will have to eat fishes that themselves eat these sources that accumulate these markers. So there is a trophic link, but the fatty acids themselves, they cannot resolve the problem. Therefore, you have to look on other tracer, and here, if you look on the stable isotope, it clearly shows you the cascade of the links between the C3 aquatic plants, some of them more than the others, how they can be used by detritivores and other groups, and themselves they are eaten by piscivores here in the top in the blue. And here look again, the C4 seems not at all to contribute, and that was groups, except maybe for this group here, for Cabaliana and Janahuaca, two varzeas that have been analyzed with fishes taken from low water and succeeding water. For one species, we were able to find it in two lakes, in Janahuaca and Cabaliana, and it was Schizodon fasciatus. And you see that the Schizodon fasciatus that our find in Janahuaca, they are segregated from the one they are from Cabaliana, in terms of fatty acid composition, but also in terms of stable isotope. And if it's clear that for Janahuaca, should be no problem to imagine, this is a biblot that have been done with the software CR, where the fractionation is already, it's already taken in account. So therefore, you got the consumers directly on the sources, and you see that the species of Schizodon fasciatus is mainly eating in cystine. And with the modeling, the CR model, it shows you clearly the importance of the cystine and other C3 plants in the food web of this Schizodon fasciatus. if you go to this was in Janawaka. If you go to Kabaliana, the group is here, and difficult to connect it completely with the same sources that we saw in the other lake, and that's because we believe that there is a mixing with other sources that maybe we didn't collect, although we collect many sources, but also there may be a little contribution that comes from the C4 plants. Of course, that has to be still to be demonstrated, but in Kabaliana compared to Janawaka, the same species doesn't eat really exactly the same food sources. So if you look in a particular fatty acids, from Kabaliana to Janawaka, the marker of, this is interesting, the marker of the plants is declining when it comes to low water, or in proceeding water it's not the case, but these fatty acids, as I said, are very important in paspalum repens. Okay, but you see that from from Kabaliana to Janawaka at the same season, there is less use of the marker of plants. Whereas if you took the microphytoplankton, the phytoplankton markers, in that particular case we have a high proportion the markers in Kabaliana compared to Janawaka, which suggests a better in Janawaka, which suggests a better food quality and availability in Janawaka. So in Janawaka, because of this polyunsaturated are more in contribution is more important compared to Kabaliana, we believe that Schizodon has a better quality food than in other lake. So I'm not gonna conclude right now because it's more or less what I said. Just keep in mind that the transfer of essential fatty acid to first consumer and potentially his hypertrophic livers can be a proxy to see what's going on also with this carbon, where it's gonna be. So you have to consider also the biology of the animals that are living in these waters and how much they could consume. The budget needs to be considered also in the total account, although it's not probably very significant in carbon budget. So we were a little bit strict by the fact that we didn't really see much about the C4 plants, and we were a bit concerned for the simple reason that the C4 plants, that is microphyte, they are very important, up to 80 tons by hectares, and if you compare to the C3 microphyte, which is up to 15 tons by hectares. So how comes that the C3 microphytes, and in terms of carbon fluxes, but also in terms of food webs, very important, but the biomass is less important in C4 microphytes. So what's going to happen with all these leaves, roots that accumulate in a particular season? And there is exception. Now, of course, when I say the microphytes are dominant in Kuruwai, it seems that this dominance is not obvious, but in other lakes, of course, they are always dominant. So what's exactly the contribution of the C4 microphyte in the food web? Are they one contribution to it? So to get there, we did this experiments. So just to repeat what I say, so what's the fate of the C4 aquatic microphyte? Question mark here. We did an experiment, degradation experiments, on field, in fact, during the cruise, in one particular cruise, where we put big tanks with the water that was taken from the Amazon, and we waited for 1 month. It was like a 1 month degradation, and I explain you exactly the protocol. So in each, of course, it was replicated, and tanks were filled with 60 liters of water. There was a control one where it was just water, and in 2 groups of tanks we put a C4 plants, paspalum repens, which was having the stabilizer, the signature of carbon of 13, and we put what we did is the biomass we put for degradation was to represent what we find in the field, which is the 80 ton by hectares, equivalent ton by hectares for paspalum. And we took Salvinia as an example of C3 plants. And in that case, we take only 100 grams to represent more or less the quantity that is available on the environment. So, and then what we did is we put 100%, the maximum that has been seen, and we consider a lower proportion to simulate a little bit what's happened at high water. We took 10% of that amount, both for paspalum repens, which is the C4 plants, and for Salvinia auriculata as a C3 plant. So there was a full percentage, which is the maximum observed on the field, and then 10%. And we did the multi-proxy analysis. In red is the data I'm going to show you now from that real experiment, and in black are the data that we did, but I'm not going to show you here. So, starting by the POC, particular organic carbon here, which was what we see is that for paspalum repens, the incubation quickly after 3 days, there is a quite quick release of the POC, and the release, of course, is much more important at high biomass than in low biomass. We quickly reach the maximum here, about 3 days, or is 6 days here. And for Salvinia auriculata, it was not significantly different from the control. So this release was not really happening there in a Salvinia compared to paspalum. And there is almost 3 times more biomass at high biomass in paspalum repens than in Salvinia. Well, the DOC here shows more or less the same thing, with the paspalum repens releasing quickly all the DOC. And of course, the release at Salvinia is not at all different from the control. Remember, control was just water that we let in the tank. Just to make sure that the temperature was not was the temperature on the board. It was not control temperature. If you look on the dissolved inorganic carbon this time, it was interesting to see that for paspalum, we can see that a high contribution of C4 derived organic matter to this total inorganic that we find. So, whereas for Salvinia is a depletion. So we have an enrichment of DIC during the paspalum repens incubation, and we have a depletion of DIC during the C3 plants degradation. Whereas that's all in high biomass. Whereas in low biomass, there was no significant difference compared to the control. So you can see that this enrichment, you have to consider how much important it is for the DIC, the dissolved inorganic carbon, the paspalum repen. So as organic, it was not very obvious, but as inorganic, we have here first glimpse about what's going to happen to this big biomass of plants. If you look on the fatty acids, we see clearly the segregation of the samples that were taken from T3 to T23, from the samples that were taken from the tanks where there was paspalum repens at high biomass, and what we discriminate is high biomass compared to all other samples is a huge bacterial biomass presenting here. And here I just showed you 2 fatty acids that can be authentic bacterial markers. There are a few others, but just these 2. And I have never seen on the literature such a huge percentage of these just 2 fatty acids, iso and anti-iso 15, and more or less the total bacteria fatty acids were about 50%, which is great. And just for this, we have here about from 10 to 30%. It shows you the bacterial increase in their activity, degrading this plant, and of course putting it into a form of inorganic. And you can see here, I would say, simple bacteria counting, and it shows you that it quickly increases. It's very high increase. In fact, indeed, if you look between 3 and 6 days, and then it continued to increase until the end, So bacteria not only are there, but data not shown here shows that they are also very active. So they are able to transfer this carbon to inorganic. And they do it, of course, there. It's present here. If you can see on the plaque, this here, most of them are present in the paspalums at high biomass, the part which is here. The other thing we measured were nutrients. So nitrogen and phosphorus, and we could see that for paspalum, a high biomass, there is a huge increase on the water. The release is done, so these plants are able to release. And we know that this nitrogen are important and limiting primary producer when they are not available in good quantities. But this degradation of this big biomass is able to release nutrients in the field. And we assume and we suggest that in one case you have this inorganic release which can be taken by microorganisms such as cyanobacteria or other phytoplankton, but also the release of nitrogen and phosphorus can feed the cystine and all the microphyte and also eventually some macrophyte if they are there in the future. This can, with that put in the yellow dot here, so the paspalum repens is not be used in the food web directly as an organic part, but what's happening during the degradation, all the release of the inorganic and of nutrients may feed the food web, the normal food web. So in that case, the question mark is less of a question mark, but more needs to do to confirm all this data, because remember, this one has been done in experimental setting. Oops. Talking of fishes, I thought interesting to show you a work that has been done by my colleague Marc Pouilly, who is a IRD guy, using data that was taken from IBAM, and he works on fishes in Amazonia. So normally there was an animation, but the format of this presentation couldn't. It was difficult to not do it like. But just the story here is to tell you that fish migrates in the Amazonian waters for all the basin. There are fishes, they can migrate to the ocean from ants, and the opposite can be the case. They can do it temporarily, they can do it for good. They are also exchanged with savanna inundated waters. So there is a dynamic of fish that can move everywhere. And what we need to know here is that sometimes it takes years to go from one point to the other. And there are important also fishes of Amazon represent 20 to 25% of the total fish in freshwater fishes in the world, which is a huge biodiversity asset. And we estimate it's counted about 5,000, a little bit more like 2,500 species known. I say known because probably there are species we don't know and others we may never know. And of course the Amazonian, sorry for the French, but Amazonian of course is impacted by human, by dams and barrage, and also by the fishing. And there is signals now that says that the fishing of this fisheries are declining, and things are not getting smoothly like we may imagine. Anyway, the fishes are impacted by anthropic activities. And one hypothesis that my colleague was working on is to try to understand, because the reason why you can explain why fishes are important to be conserved is how to trace these fishes, how to explain that the connectivity of the water is an important aspect of the ecology of the Amazonian waters. So he implemented a method, and first of all he looked at the ratio of strontium 87 and about strontium 86, which is known to characterize different waters. The scale of the difference between the strontium 87 and 86 is very small. It's it starts from 0704 to 0744, so 45. It's not that big. And for example, he is able to identify each type of water. Here he gives some examples. Each water can be clearly identified by its strontium ratio. And the question was, as we know that strontium can be assimilated to some extent by organisms, especially organisms that have calcitic in their physiology. He had this hypothesis to look on this on the fish. Are the fish able to get a signature of the water where they live? And there were works that have been done previously that shows that otoliths can integrate in their aragonite. They can integrate this strontium, and I'll explain you how. However, to get that, the fish, you have to kill it. It's very complicated to extract the otolith. So you kill the fish, get the otolith. It's quite hard job to go there and get it. But when you get it, you get your otoliths, and the otolith, what is it? Is this little bone piece that is just behind the eyes of the animal, and in which you can see streaks, all these marks, and each marks can represent a season, a year, and it implements each year. There is each season or year you can see it implemented in these marks. He decided to look on another part of the fish, which is the I have a blank. I can't translate to you. How you say in Portuguese, écaille? Sorry? Gills, not gills. No, you know, that's on the skin. The skin of the fish, the scale. Voila. Oh, thanks very much. I forgot about the word. The scale. So, another interesting part is this analysis is not lethal. You don't kill the fish. You just catch it, take it. So he has to confirm that this strontium can be a good proxy for the water where they live and the quality of the water too. And here are the results. So he took two fishes, one which is herbivorous, one is carnivorous, including the Schizodon fasciatus that I talked to you about earlier. And amazingly, that what we see here is the biplot of the signature of the water in where they were fished, with the signatures. Yeah, there is two. You look, there is two lines. One are considering the otolith here in red, and the other one is the scale on green, and the air square is quite gray. They're almost similar, and each one is relying on the water in where they were fished. The fish that were, you can see that each water has a different signatures, and the fish gets exactly where they should be. So wherever you take otoliths or scale, you can trace the fish in the water he was getting. And I will show you why this application can be interesting. For instance, if he took some fishes from the water where they were where they were fished, looked on the water. Here the water is in blue. This is the ratio of their, in this time, the scale, not the otolith of the fish. And also he went to the market of the village around or the city around, and he looked on the proxy on the strontium of these fishes, the same species. And I believe this is Schizodon. And for example, if you look in Beni Savan, that's clearly the fish comes from the water, from the lake where it is there. In Itinez, now he didn't make a commercial, but if you go to Beni Rivier, the river from the lakes there, it comes from somewhere else. And Tapajos, you can see that the Oplias, the fish Oplias, was really from the water, but the Schizodon they were selling was not coming from that. It came from somewhere else. So, who knows? Maybe sometimes we need to trace this for good reason. So it would be a good option. It's a really great job that will be published very soon, and it's quite amazing. I thought since it has been applied for the first time in Brazil, I thought it would be good for you to know about it. I believe I have passed my 45 minutes, but I want really to show you one aspect, because we talked with students yesterday and the day before, and I kept saying about this idea of having a multipraxy to trace organic matter and what can be the interest. So let's go back to France, in a region which is not tropical, very temperate, in coastal areas in the north of France, and we have zones that are very much used by aquaculture. And big question, all the stable isotope studies and other tracer and even gut contents things, whatever name it, they were really having trouble to understand where the food source really came for to sustain the huge aquaculture production that is there. And why the question was there, because in these waters, plankton, which is usually, we know it in south of France, marine d'Oléron, phytoplankton, mainly diatoms, are the ones that sustain this big production of aquaculture. It was not the case there. But what we can see by eyes for the people that study the places is that there is many brown algae. And brown algae is an issue because, first of all, they were a little bit far from the production site, but brown algae is known also not to degrade easily, and we cannot see how it comes, because diatoms, it's simple. You have your production of aquaculture. We're talking about conchiciliculture, which means bivalves. They just filter. So how do we imagine brown algae intervening there? And we decided to look a bit deeply, thoroughly this, and also because many of the study has been done usually in one part of the day, most convenient for field work, wherever it could be. So we decided to make a cycle, a tidal cycle there, and we took SPOM. And we looked on the tracer, and we did it in 2 seasons, in winter and in summer. Now, as you can imagine, and here what we done is just looking at the fatty acids, and from high water to low water here, you see that the tracing, we are talking about a cycle of 12 hours here, from F1 to F7, that's about even 12 hours. From these 12 hours, the composition of the organic matter, it's a cycle, changed drastically. And it changed from a dominance of bacterial markers at the high water. And then we get, I hope you can see the colors, we see the contribution of the phytoplankton here as a second part of the cycle. And then we get macroalgae, brown algae at low tide, coming in the water because it flushes from the region where there is the brown algae, and then get back again to some phytoplankton and some seagrasses, and then again to high tide, back to the bacterial dominancy, pro-dominancy. So what happened is, this vision of the cycle give you another the idea about the dynamic. You don't decide at 1 hour of the day or 2 that this is the dominance of the quality of the organic matter. And we are talking about quality, it's important. Some polyunsaturated can be present only at one part of the day. And not surprisingly, we saw that one species of bivalve we did 3 species of bivalve, but one of them, she filters always 2 hours before low tide, whereas the other species will be filtering 2 hours after low tide. And it's because they know exactly when they get their phytoplankton, seagrasses, and brown algae. So the animal is not stupid. He waits the right moment, the water comes, and then even the water continue to fall or to grow, they stop to filter because they get what they want at the right moment. Voilà. So I think you are all tired. I'm going to stop here. I had another example about this, but yes. Thank you very much for your listening. Thank you. Just to mention to the people that are not in this room that they can also ask questions. It seems that you can click in a little icon on the left side, which is in Portuguese levantar mão, if I understood. Ah. Move, raise your hand. Right.
Can you hear me?
Yes, I can.
Okay. First of all, thank you very much for the presentation. It was very, very interesting. A lot of new information for us packed together. So it's a nice exercise for the brain as well to think about so many new stuff for us at the same time. Right in the beginning of your presentation, I got a question about methodology
Okay.
issues. You talked in the beginning about the end members you sampled for isotopes and also fatty acids, I guess. And then you mentioned that, or I saw on the slide, that you used for phytoplankton, you used the zooplankton signal, right? Because it represents the phytoplankton.
I use just the phytoplankton signature, not the zooplankton.
Phytoplankton. Ah, okay. But
Also, we did analyze zooplankton. I just didn't show you the results, but we did. It's a signature of the phytoplankton.
so you got only phytoplankton signal. And for that, did you use the plankton, a phytoplankton net and concentrate it?
And then we filtered. And then we filtered, of course, with the appropriate filter to get the phytoplankton part of it.
My question is because I can see in the picture you use that there is a very like green material inside the net, but when we work in very oligotrophic places, we used to have a lot of detritus mixed with the phytoplankton in the net. Do you know what kind of methods can be used in that case? Very turbid waters, for example.
Mm. It's a very good question. When in turbid water, how to remove the planktonic part from the well, you can guess it by the tracer, because what you have to understand is that anything detritic, in terms of tracer, if you know the panel of signature of stable isotope, for example, you can know exactly what's the contribution of the detritic part compared to the living part. Okay, but for the fatty acids, it's much easier for the simple reason that detritic part has more or less no polyunsaturated, yeah, or very small amount. So in case you've got like imagine a 20% of one particular polyunsaturated fatty acid, you can be sure that it's really the predominance from this microalgae or cyanobacteria or so on. So technically, I believe that the best we can do, we have done it in terms of separation, in terms of filtration. But I cannot imagine myself on the field, of course, doing better than that.
Yeah, because my question is mainly because of carbon isotopes. That's what I was struggling with. Oh, sorry.
Mm.
I was struggling pretty much with the carbon stable isotopes from phytoplankton. And then if I want to trace how much of the phytoplankton I can find in the sediment,
Yes.
I should really separate it from the detritus that is
Alors,
mixed with the terrestrial sources. Uh,
from CO2, but in temperate, I have never done it in, of course, in in tropical climatic and in Varzeas. But in estuarian studies, we do use a small, comment dire, biofilm. That for to migrate the phytoplankton that is on the sediment, but it's at low tide, of course. There is no water. So we go there and we make a migration of all the microphytobenthos towards. It's a rough estimation, but it's a better estimation than to take the sediment itself anyway. Voila. That can be done, for example, in places like this. But of course, underwater, it's just impossible to imagine doing this.
it's tricky.
Yeah.
Thanks. And the other thing is because I don't know a lot about fatty acids and how it's used. You also showed that the fatty acids they change a lot. If I'm not wrong, is the fatty acids content, or I don't know if it's also the composition. They
Yes.
change a lot in the tissue of the plants over time, but actually with the flood post, right?
Yes.
What what makes this change in the tissue of macrophytes, for example, or plants?
Remember, fatty acids, it's physiology, okay? So it can be impacted by climate, temperature, by the plant is, if it's a plant, for example, is it how the age of the plant can change the composition, quantity and quality. Uh, Again, that's the main things I can see. Also, we have seen, for example, that some plants, when they are eaten, it's like the cow when it eats the grass grow more. For example, a pyréphyton, which is this microalgae that live in the rocks, more you eat them, more they produce. So in quality they're better, in quantity sometimes better too. So, but you, of course, when you analyze that, you have to normalize the analyze to the exact weight to compare, of course.
Because you also compare the end members, the fatty acids in the end members. Let's say if I want to take a sediment sample
Hmm.
and tell what composition, what the sources are of organic matter to the sediment sample.
Yes.
Then if I take the end members, but I take it only once, because the sediment integrates it over time, right?
Yes. Yes. Mm.
So is it a problem if I take the end members only once? Or the main fatty acids to identify the different sources will always be there, regardless of the time of the year?
Want to compare, in my opinion, the best way is not to focus. For example, sediments is more or less you have. If you want to see the change on over time, is really to look on the whole composition, and wherever you get, and then you have pulses of organic matter. So it's all about when exactly are you going, how many times you're going to sample. Is it every day or every hours, or it's up to you. For sure, if you, it's the way how the sampling is happening that will give you the answer to your question. So if you sample too much between too distance between two sampling time, you may pass the dynamical aspect of what's happening in the sediment. It can be possible. But just to make sure about something, fatty acids give you a signal of about the last month, no more. Okay, no more than that. If you go more in details, for example, if you look on phospholipids of some consumers, if we talk about the consumers, then you can have something about the last week. Okay, that's if you look on the whole composition. But I want to understand something. There are geologists and geochemists guy. They look on fatty acids in the earth, with a little come on, pensare yeah, with coarse things, and they can trace the history of the sediments for 100,000 years with fatty acids. Okay, but they are not the same one. I'm talking about saturated one, the ratio of saturated. But polyunsaturated have a very quick dynamic. So 1 week, 1 month, depend where, and never. This is one thing I learned by time. Never conclude by looking what's happening somewhere else. You really have to make your mind about your environment. For example, the marker we have used here for to trace phytoplankton is not the one we are using, for example, in coastal science, because we had to deal with a typical phytoplankton. Okay, it's not the same. So the markers were a little bit different. So you have to focus on you have to have the source, to define them, and then quickly see how they change within the sediment or anything else.
Thank you very much.
Yes. Yes, no, it's okay. No, might I hear you from the room, but I guess the people not in the microphone. Take another, take another one for someone else.
Estão vendo?
Yes. Mhm.
What about now?
Yes. Okay.
That's the proof that I'm old-fashioned guy. Even I try to be on time with the technology have always had problem. So again, thank you very many times for both of you, Tarik and Gwen, to be here, and it's really a pleasure for us, and, especially for me because I'm fan of both of you, and I always learn many things. Last week we had here, no, first to say that in some moment here I counted uh, some about 18 labs. So for all the people there in Brazil, thank you for listening us, and thank you for being in this classroom. It's great, and I hope we just starting these ideas, and I hope to get many feedbacks from the guys in different labs, and we have to improve, and also not just to have Juiz de Fora as a hubby, the main hubby for this kind of conference, but we in different labs in Brazil we can have this and share experience and speakers with us. So now my question actually is not a question, is as a Brazilian and a limnologist, I can see realize how many ways and tools we have to evaluate the problems that we are experiencing in Brazil in this last 2, 3 decades and in ahead. So put together the Gwen and the Tarik talk together in the same package. Wetlands plus fat acids as tracers for one side. The other side we have Brazilian, in terms of frontiers in limnology, we have dams for everywhere in different sizes, in different watersheds, different biomes, and for our understanding, we are not stopping to building dams at all. And also we have a very strong land use because the economy is growing and depends on the not just mining, vegetables, soybeans, and many other things. And also the third one, it's not just our responsibility, but we respond in many different ways for climate change. And I'd like to hear, Tarik, from you some idea, because I'm now during these last 2 hours, I was thinking if someone from the sky come to this room and said, okay, we have dams, we have land use, we have deforestation slash land use, and we have climate change. So you guys have as money as you can to build a project, to put all the efforts together, and that's I'd like to hear a bit from Tarik, and after from Gwen, for example, to design a project, a kind of effort that we should have for these 10 years in ahead, for example, to have students that will be prepared to fight or to improve science for example, in 2025.
Okay.
For using this idea, we could have, for example, 5,000 PhD working in solving problems, not just applied problems, but of course theoretical and conceptual problems. If someone gives us
That money.
the opportunity to have in 20,000 to 5, some kind of thousands, 2, 3, I said 5, but maybe it's too much, spread in the country and connected with different labs in the world and trying to to solve the problems. I know it's a kind of
Difficult question.
question for a bar, not for
Yeah.
a
No,
like this, but
in fact, this classroom is a way of doing what you are saying. Let me explain. I'm not. Amazonia is, of course, some place where I learn. I love to work in, but it's not my main topic, and it's not the main topic of my studies in general, since I am a researcher. So my main topic is mainly done about coastal science and tidal, mainly mangrove ecosystems. I can have an answer to you about these mangrove ecosystems to your question. It will be much more difficult for me to imagine solutions for, I mean, although there are few solutions that everyone knows, we could stop everything and start back again, but it's not possible. For example, for mangroves, and these are also a kind of tropical forest, it's too late in many ways. It's too late to save what you could imagine being the perfect ecosystem that you don't, the native ecosystem. There is so much harm done to that ecosystem that it will be not possible. And so the people who are thinking now, they cannot even think about saying, okay, even if we have all the money of the world, how can we go back to before or save whatever it can be saved? Problem is now they are more thinking how to save witness of what can be, what was the native situation, witnessing. So it's a little bit pity, and the harm is continuous, and I guess it's the same for Amazonia. But when you start asking a question, the first thing came to my mind is if you have to put money somewhere. In my opinion, is maybe not only in research, and maybe not the first part to do is in research, is to educate people. It's to explain people without being an extremist ecologist, which I am not, is really to educate people to understand that they are simply destroying part of their asset for the future. So, and if this message can reach a maximum of people, especially the people they live in these places that are endangered, I believe this money will be well used, of course. And then, of course, we can look this example of strontium that I gave you that didn't cost that much. I mean, in terms of, you have to imagine, it's not costly, okay? It's just time consuming, of course. A lot of, you need, but these are kind of things that science can reach, give you proxies, I would say, skills to explain the way how it can be monitored. But science is not able to do politics in reality. To say, okay, we will be able to resolve the problem. Science is not going to stop the construction of the dams. Science is not going to stop the deforestation. We can just say, if you continue to do so, this is good things that can happen. Maybe also good things, maybe. Some places it can be. Also, there are bad things. So that's much I can say about your answer. Well, if there is somewhere to invest is in education. Education of every strat of the population, especially those they live in these places. And maybe Gwen want to add something.
Tiens.
well, uh, I think it's much easier to ask the question than to answer to the question. It is very large question, and I would say I will answer in three. Well, I will start saying that if the I would start saying that it is difficult for me to remain optimistic as a scientist personally. This is the first point. Because we are the first witness of what's happening. We know what's happening before the rest of the population, and we have little impact on preservation in general. And sometimes I wonder if I should put more energy to go down to to inform or educate, as Tarik said, the population, and instead of putting more energy on doing science. And in fact, to be honest, this is difficult for me to take this decision. This is the first point. But when I believe in transmission, and I believe in education. Tarik said basic education, but I think high-level education is also very important. And the more scientists we can educate, or the more we can educate, the more chance we have in the future we will have to preserve the environment in general, and Amazonia also. Then, what type of science would be the best? You know, science, for this environmental science in general, we need to cope with various approaches. Of course, we have so many science-specific specialty. We need so many specialty to solve a general problem that we have to join people from different places and from different topics. And there is field work necessary. Of course, monitoring is necessary. Remote sensing, modeling, and so on. Experimental science, experimentation, and all of this is necessary. But I would like to say to the young scientists that are entering in the career, don't forget the field. I think the field work and the real physical contact with the nature that you are studying, this will give you more ideas, and this will make think you more maybe than theoretical stuff, because most of the time you are, if you read the literature, etc., they are all in a given paradigm, and to change the concept, to change to say no, this is not how the system is working. In fact, the system is working another way, and this is very important. For be able to do that, you need the contact with the nature that you are studying. That's very important. Otherwise, you can say wrong things. You can have wrong conclusions just because you were not at the right moment to observe the very important process at the right place at the right moment.
I have a question, and it's related to the graph where you compare the scale and the other part of the fish. And I can see in the graph that the up part of the graph, the lines are a little bit separated. And my question is why it happened and why it happened also in Tapajós, because I remember the other graph you present just in front, the the values for the scale and for the other part of the fish, they were very different. And why it happens in Tapajós?
So that would be so that in this one. Sorry. You're looking for this comparison. This Tapajós is not made here. You agree with me?
No,
Yeah.
Tapajós is the next one. In the next graph, there is information about Tapajós.
This one, this Tapajós. Yeah.
Oh, this is not okay.
Voila.
Okay,
This is two species.
Okay, I got you now.
Yeah,
But
this
the
is-
first one, the previous graph, the up part of the graph.
Yeah, there is a divergency. I asked the same question to my colleague Marc Pouy because I said to him, if you want me to present carefully your stick, I asked him that question. And he mentioned that this divergence is very small. I mean, the most important is the R square. Because for each, the R square, as you can see, is
Nice.
it's perfect.
And it's amazing because when you analyze the scale, you don't need to kill the fish.
To kill the fish. Exactly.
That's
And it has never been done. And he had this brilliant idea. He told me one time he was having a talk, drinking a beer in Manaus with some people, and he said, "Why not try?" Because they were talking about the otolith. This method is famous for otoliths. It exists. People know about it. But he say, okay, he knows the chemistry. We are talking about. Remember, we are talking about the substitution. The water, you have this ratio of calcium and strontium, and then the otolith, when it grows, the otolith and the scale grow because the animal grow with the season, so they need to take the calcium from the water. But not only they take the calcium from the water, but they also take from of the strontium too. Yeah.
And I got you. And my next question is, how many years of information you can take from the scale? As
This is
old as the fish?
it's quite immediate, I would say, because you know what's gonna happen. You are gonna look on the composition of one line,
Okay.
so you can follow the fish. You go back to the line and decrement, so you can know his migration from the original water, say in the Bolivian Amazonia, and how it goes until the Obidos, for example.
Okay.
You just follow the links, the listri, the line, and each one will have its own characteristic. And this is exactly what he's doing right now.
Gotcha. Thank you for your presentation. It was amazing, and I think we have a lot of things to
Of
to
course.
start a cooperation,
My
and
pleasure.
all this analysis will be awesome. Thank you again. And if someone doesn't have question, we can finish now. And yeah, thank you for both of you.
Thank you very much. Thank you all.